Today in this lecture, we are going to talk about the role of distal tubule, especially the early part of distal tubule in urine formation or the mechanism of urine formation. So the role of distal tubule. Now we have discussed so many times that urine formation begins with filtration in the glomerulus from the glomerular capillaries into the Bowman's capsule, and then the movement of the fluid through the proximal tubule, where a lot of reabsorption of fluid occurs from the proximal tubule, then the movement of fluid through the descending loop of Henle, then the thin ascending loop of Henle, then the thick ascending loop of Henle, and finally it reaches here. We have discussed all the steps in detail, but as we know that when we discussed the loop of Henle, we discussed that the first part of the loop of Henle was allowing the reabsorption of water, but it was not allowing the reabsorption of sodium chloride, etc. The ions were not reabsorbed, but water was reabsorbed. And in the thick ascending segment of the loop of Henle, there was no reabsorption of water, but there was a reabsorption of ions like sodium chloride and potassium. So the same phenomenon occur in the early part of the distal tubule, in which all the electrolytes like sodium chloride and potassium, they get reabsorbed, but water is not reabsorbed. So water remains in this tubule, water remain in this lumen. It is not reabsorbed, but the sodium chloride and potassium, they get reabsorbed. Now this is one function of the distal tubule. The very initial part of the distal tubule is in close proximity with the juxtaglomerular apparatus. As we discussed previously in a bird's eye view, we discussed that this is the nephron, this is the Bowman's capsule, here we have the glomerular capillaries, here is the proximal tubule, there is a loop of Henle, then the distal tubule. But in the initial portion of the distal tubule, we have some cells, the macula densa cells, which are basically forming the juxtaglomerular complex. Now this part of the distal tubule, which is in close proximity with the juxtaglomerular complex, it helps in the maintenance, increasing and decreasing the glomerular filtration rate, the rate at which fluid will be filtered into the Bowman's capsule, and also it will increase or decrease blood through the glomerular capillaries. How it does this? Here we have some special cells, the macula densa cells, which keeps on detecting the concentration of sodium in the tubule. When the concentration of sodium in the tubule increases or decreases, the macula densa cell senses this thing, and it, through the help of juxtaglomerular complex, it leads to release of renin. It either increases the release of renin or decreases the release of renin, which ultimately leads to increase in arterial pressure or decrease in arterial pressure, both in the afferent arteriole and the efferent arteriole. The afferent and efferent arteriole were discussed in great detail previously. Now that's one thing. The distal tubule is increasing and decreasing. It is helping in increasing and decreasing the glomerular filtration rate and decreasing the blood flow through the glomerular capillaries. And it is also helping in the reabsorption of ions like sodium chloride and potassium, but it is not reabsorbing water. So this early part of the distal tubule, it is also known as the diluting segment. It is the diluting segment because when water is not reabsorbed and water remains in this portion, it dilutes the urine. So it is the diluting segment. Now how sodium chloride, potassium are reabsorbed? Here the cells in the distal tubule have a sodium potassium pump, and this sodium potassium pump is throwing out the sodium from the cells outside the basolateral membrane, and it is throwing potassium inside the cells. When the sodium is thrown out, there is deficiency of sodium, and sodium starts entering from the lumen. Now this is the tubule lumen, this is the lumen. Now sodium is thrown outside through the basolateral membrane. This is the basolateral membrane, and here we have the tubular luminal membrane. From the luminal membrane, the sodium enters the cell because there is less sodium. The sodium has been thrown out by the sodium potassium pump. Now when sodium enters, this sodium is followed by chloride, and this sodium is then followed by chloride. But the entry of sodium and chloride is with the help of this symphotor known as the sodium chloride channel. Now the clinical importance of the diluting segment or the early distal segment of the nephron is that this sodium chloride channel can be blocked with the help of thiazide diuretics. So the role of thiazide diuretics is that they block the sodium chloride channel. Now sodium chloride cannot be reabsorbed from the lumen. Sodium will be thrown out of the basolateral membrane, but sodium cannot be reabsorbed from the luminal membrane, so sodium will go out in the urine, and this will cause diuresis, and that's how thiazide diuretics works. So the functions of the early part of the distal tubule are the reabsorption of sodium chloride and potassium, and no reabsorption of water. So it is acting as a diluting segment. And another thing is there's some special cells in the early part of the distal tubule. With the help of GG apparatus, or as a part of the GG apparatus, the juxtaglomerular apparatus helps in increasing and decreasing the glomerular filtration rate, as well as the blood flow to the glomerular capillaries. So that's all about the early part of the distal tubule. Thanks a lot for watching the video.